Great. Thank you for the introduction. Today I'm going to talk about slightly different solution for the wood engineering. So we're going to use the genome editing or genome engineering method to engineer wood. So the CRISPR, if you ever heard of CRISPR technology, so it's can be widely used for genome editing in both animals, mammalian cells, or plants. So the CRISPR stands for cluster regularly interspace short palindromic repeats, which was first discovered as a immune system in bacteria. But later on, it was first demonstrated as a can be used for genome editing by these two great researchers, which is are Jennifer Donna and Emmanuel Charpentier. So later on in 2020, both of them were awarded for the Nobel Prize in Chemistry based on their discovery for the CRISPR technology in genome editing. So as of 2024, there have been a few genome edited products produced using CRISPR technology. For example, the non-browning lettuce, avocado, and bitterless mustard green, and also high GABA tomato. So besides the crop products, so the CRISPR technology or genome editing technology can also empower the tree bioengineering or wood engineering for a sustainable future. So here, for example, in the aspects like the pulp paper production, engineer wood production, or biofuel production, or even the carbon sequestration here. So to achieve the highly efficient genome editing in the woody plant or the tree, so we have developed a high efficient transformation system in poplar, and which allows us to get the like transgenic poplar or genome edited poplar in just 3 months, and then we can use that for greenhouse test or even in the field. So in 2018, Bean's Lab already published the paper to produce the high-strength densified wood, and also known as superwood. And so basically, they used the natural wood, and first did a chemical treatment to remove the lignin, and then followed by hot pressing for the superwood production. So, but there's some still some cost concerns about this technology. For example, the high energy consumption or chemical intensive processing, and also some chemical waste as well. So then we proposed to use the genome editing tool to just reduce the lignin in the tree or in the wood. Then we can skip this chemical treatment and directly proceed to the hot pressing for the superwood production. And so in this way, we expect low energy consumption, a chemical-free processing, and also no waste generation. So to achieve the lignin reduction in poplar wood, so first we look at the lignin biosynthesis pathway in poplar, and we expect to target the one of the most important like enzyme like 4CL, which is like the first step of the enzyme for the lignin production. And then we decide to the target site to just target this enzyme or this gene. The technology is to introduce a premature stop codon to stop the enzyme or the protein translation. So in this case, the 4CL1 will not make a functional protein, so we can make it a knockout mutant for this enzyme. So and our the DNA sequence data just show if there's successful premature stop codon or mutation inserted at the target site. So then we put the genome edit the poplar in the greenhouse for 6 months, and then we look at the biomass. There's no growth penalty in the greenhouse for 6 months growth. And then we just peel the wood, the bark, and we look at the wood. Basically, the genome edited wood actually can show the red color red wood, which is a consistent phenotype for reduced lignin there. And then we just collaborate with Bin's lab and to do more like the wood analysis. So first we look at the lignin content. So if you compare with the wild type wood or natural wood, poplar wood, actually, our genome edited wood actually had a 12.8% lignin reduction, but there's no significant changes in terms of cellulose and also hemicellulose content. So then we further look at like how about this, like if you use the genome edited wood to process the densified wood. So now we use both the natural wood and also the genome edited wood to look at their strength characteristics. So and we can see here the non-densified wood compared with the non-densified wood, actually densified the genome edited wood can have a over like 5 folds strength and intensity, which is comparable with the densified wood, which used the chemical treatment, like the original protocol from the superwood procedure. And then we further look at the density after the pressing, and you can look at you can see that the densified genome edited wood actually showed slightly higher like density compared with the densified wild type wood. And besides the lignin content, we also test or look at the lignin structure and also distribution. And we can see here, sorry. ABCD shows like the especially the C and D shows there's no significant like the lignin different at the cell wall location. But if you look at the cell corner, actually the gene modified would actually shows less lignin content here. And also there's, if you look at the NMR data here, actually, there's slightly difference in terms of, for example, right here, like there's no, it's like lower in the genome modified wood here. So lastly, we further look at like the texture of fabrication, like the characteristic of this genome modified wood. After soaking in the water, we found like the genome modified wood actually is quite softer than the wild type wood or natural wood, and also you can see, although the genome modified wood actually is have less stress compared with the wild type wood before the pressing, but after the pressing, the densified genome modified wood actually is quite higher, have a higher strength compared with the densified wild type wood. So another question maybe like, will the genome modified wood tree can be regulated as a GMO? Like to give a no answer. So actually, we recently just developed the transgene-free genome editing system in poplar. So similarly, but in this case, we just select the transgene-free genome editing poplar instead of like transgenic plant. So in this case, we can generate the any like transgene-free genome editing plant, which can just bypass the regulation effect. So our current research direction is focused besides like the reduced lignin content, and also we focus on the accelerated like the growth and biomass production, and also improve like the fire resistance and also lignin structure like the reprogramming. So yeah, we are open to any collaboration. If you think like that you are not satisfied with the natural wood, so want some specific characteristic, we can keep connected to better figure out like what can we do like using genome editing tools to like DIY the wood. Finally, I want to thank my postdoc supervisor like Yiping Chi, and also our collaborator like Dr. Coleman, Dr. Hu, and Dr. Liu, Dr. Mao, and Dr. Kim, and Dr. Pan for their contribution to this project. I would like to take any question you may have. Thank you.
Thank you very much. Question? Yes.
You mentioned there's no growth penalty. Thanks. There's no growth penalty in greenhouse. I'm wondering if there is like a long-term potential growth penalty if you move the trees into real world, real natural conditions, and when the trees get bigger, how is the performance of the GMO trees compared to the natural trees? Yeah.
Yes. The field testing is right now is in processing in our field, and also in terms of growth penalty, it also depends on how much lignin you were reduced. If, for example, there's another publication, they did a more severe, like I would say, lignin reduction. For example, 40% lignin reduction. Then you will see even in the greenhouse, the plants saw like dramatic, very sad, growth. So I think how much lignin reduction is quite, important for, affecting growth effects. Yeah. Yes.
Do you have any idea where the lignin is reduced? Is it cellular corners or is it uniformly?
Yes. So if you look at the images there, so we didn't see significant like the lignin reduction at the cell wall, but more like the cell corner. I was just like maybe there's a cell wall structure because like in the corner there's much thinner like the tissue. So I would say like if there more lignin accumulated there, so but when you remove the or knock out the enzyme, so like maybe the lignin is much more reduced in that lignin enriched area. Yeah, that's kind of my guess. Yeah, sure. Yes.
Sorry, also, yeah, both we can go back to your chef.
What's the current regulation that's the United States government about growing genetic modified or edited tree species?
Yeah, it's quite a tricky question because like usually we're talking about GMO, right? Genome modified or gene modified or genome modified, organism. But as I mentioned, like because using genome editing tools, we can just we change the DNA base without introducing any external DNA or the genes, right? As I mentioned, like the new protocol we just identified. Basically, in that protocol, we can just basically make a mutation without introducing any external genes from other species. So it will not be regulated as GMO. So we will not regulated by the agency. Yeah.
And final question, Yoshi.
Yeah. So when you compare the wild type and the edited one,
yes,
do you see the difference in density of the wood, or because I thought the cell would look thinner, or maybe that's the part.
Yeah, right. So actually, I think we didn't look at the intensity from the before the pressing. Yeah.
So this is I think an important parameter, because if the apparent density is smaller, it's easier. I mean, if the lumen is bigger and the wall is thinner,
Yeah.
compress
Yeah, my guess is like the density, the genome modified density is like it should be smaller, yeah, lower.
That would explain
Yeah,
that you have higher density.
after pressing. Yeah.
Yeah, and one thing I think you need to look at is the difference in the growth rate also after this genetic modification, because with just one type of wood, depending on how fast they grow, the density will be different. Right?
Yeah.
So,
Good question.
okay, great. Thank you very
Thank you
much.
so much.